Hey guys, so this is the PowerPoint presentation on meiosis, which is the unit that we're covering for this week. And it is the process that is used to create your gametes, which are, as we learned from yesterday, your cells that you use for reproduction, like your sperm and your egg. So, here we go, meiosis, which is similar to mitosis. It sounds similar, right? It sounds meiosis, mitosis. They're easily confused, and that's good and bad because they're both the process of cell division, but they achieve different functions, or they achieve different goals. So in meiosis, it's defined as the process where the chromosome number in a cell is reduced by half. And we've discussed why this is important, why your gametes would need to have half the chromosomes. You make your gametes by this process. Those are your sperm and your egg. Sperm if you're guys, eggs if you're girls. And the end result is 4 haploid cells. And we went over the word haploid today in class when we took our notes. Remember, it is a cell that has half the normal number of chromosomes in it. So the end result is that you have 4 cells with only half the chromosomes that a normal cell has. So it begins with interphase, and this interphase is the same as it is in mitosis. It's G1, and G2. Remember, we learned about the cell cycle. So interphase of meiosis, G1, growth, normal stuff happens. S, you copy your DNA. And G2, more growth and normal function, but you also are double checking the DNA to make sure you're good to go. So after interphase comes, well, synthesis is the process of copying your DNA. So chromatin strands get duplicated in S phase. Okay? And then you go into prophase. And prophase 1 is very similar to prophase 1 of prophase of mitosis, but there are some subtle differences. So it's similar, except for this thing that happens called synapsis, which is when 2 chromosomes that will pair together, and they'll do something funky. Ready? There's mom and dad's chromosome. They're going to get together. This is called synapsis. And watch what happens. Once they're combined like this, they make something called a tetrad. So that right there that you're looking at is called a tetrad. They will do something called crossing over, which is when they exchange genes. That's like if I were to shake a hand with somebody else, and then when we pulled our hands away, I had their hand and they had my hand. We've exchanged chunks. So now I am mostly Mr. Ahern, but I have pieces of them, and vice versa. So you can see this in the image. Right here, we have 2 individual homologous chromosomes. Here in synapsis, they are getting together, they are crossing over, and then here you can see when they get pulled apart, they have a little bit of each in them. And let's just to go back, this is very important. This creates diversity. This is why you look different from your siblings, because your chromosomes that you got from mom and dad are just a little bit different than the ones your sister or brother got. So, early prophase, very similar to mitosis. Chromosomes form, loose DNA condenses. Okay? Now remember, you got half of your genes from mom and half from dad. So here's dad's second chromosome. Let's find mom's. Well, there's mom's, and we can tell they're similar because of their color. The nucleus dissolves, just like in mitosis. They will get together through synapsis. There we go. And they form a tetrad. Now, this is the big difference. This doesn't happen in mitosis. In mitosis, they just line up above each other. They do not get together to make a tetrad in mitosis. So meiosis is where we see this. And when they get together, they do their crossing over. Everything else is very similar to mitosis. Metaphase 1, what do you think happens? Those pairs line up along the center of the cell. There they go. Anaphase 1, well, what happens in anaphase of mitosis? They get pulled apart. So there they go, to either side of the cell. So the homologous chromosomes get pulled apart, but I still have sister chromatids, right? In mitosis, I have half of these guys. Well, this is where I would finish if it were mitosis. But remember, meiosis, we do it twice. So we're going to do all of that again. Telophase, just like in mitosis, the cytokinesis will start to happen. You'll see the cytoplasm beginning to pinch in the cleavage furrow if you're an animal, or a cell plate if you're a plant. The nucleus will reappear, and there you go. So this is important. Between the first meiosis division and the second, there is no interphase. Because remember, in interphase, you copy your DNA. And we don't want to copy DNA again, because that would mean we'd end up with cells that were still diploid. So you have to skip interphase 2 so that you can actually get down to half the chromosomes. That's very important. So remember, you got to reduce the amount of your chromosomes by half. If interphase 2 were to happen, you would copy your DNA again, and you would just have cells that were still diploid. So we don't do that. They do not divide again. The chromosomes do not copy again. So, telophase 1 finishes, and you go right into prophase 2. This is the second division in meiosis. So here we go, prophase 2. We know it's the second division because look at that 2 right there. There it is, okay? So in prophase 2, same thing as in prophase of mitosis, the nuclear envelope disappears. No new DNA was created. So there goes the nucleus. It's disappearing. Spindle fibers form and all that, moves them around. Metaphase 2, just like in mitosis, they get pulled to the center of the cell. And then comes anaphase 2, same as mitosis. These sister chromatids get pulled apart to the opposite ends. There they go. And then telophase 2, it is identical to mitosis again. Cytokinesis splits those cells, but this time we have 4. We had 2 originally, and then they divided again, and now look, we have 4 cells. The nucleus reforms. The chromatids uncoil back into loose DNA, and the end result is 4 haploid cells. These could be 4 sperm cells, right here, sperm 1, 2, 3, and 4. So 1 cell becomes 4 in meiosis. In mitosis, 1 cell becomes 2. So it's different making sperm from making eggs. Spermatogenesis, the word gen, whenever you see gen in any word, it means beginning or to make. So like if you think the thing that comes first to mind, I think Genesis in the Bible, and that is the creation of earth, right? So gen means to create. Sperm, well, you know what that is. So spermatogenesis is the creation of sperm. It happens in the male testes. You do meiosis 1 to make 2, meiosis 2 to make 4. You get your 4 sperm cells. There they are. You plug some tails in there, and they're good to go. Those little puppies might go fertilize an egg now. So you make 4 at a time. This is not the same for oogenesis. Ooo means egg in Greek. Genesis means to make. So this is the process of making your eggs, ladies. So oogenesis is different. You don't start with 1 and create 4. You start with 1, you end up with 4, but only 1 is going to be the one that you will use to make a baby. During meiosis 1, just like in normal, cytoplasm divides. But look, you guys see this? It divided inequally. This one is called a polar body. It's small and useless and won't be used for fertilization. This one is big and healthy and will go on to meiosis 2. That divides again, and now I have 3 polar bodies. These 3 are useless. They're rubbish. They're going to go in the trash. This one could be fertilized. Now, why do you think it would be important to only create 1 big egg rather than 4 medium-sized eggs? We'll see. So big differences between mitosis and meiosis. Here are things we need to know. Mitosis, you divided your somatic cells. Those are your body cells. In meiosis, you divide to form your gametes. That's your sex cells. An example of a somatic cell is your blood cells, skin, nerve, eye, hair cells. Those are all somatic cells. Meiosis, your gametes, are only going to be your sperm and egg. You end up in mitosis with two identical diploid cells. Diploid means you have all 46 chromosomes. Identical means you're the same. In meiosis, you end up with 4 unique haploid cells. 4 unique haploid, half the chromosomes cells. Importance replaces your aging cells in mitosis, right? You get a cut, you need to replace those damaged skin cells, so you're going to do mitosis to replace them. In meiosis, the importance is only to make your gametes, make your sperm and egg cells for fertilization. So review. You guys can do this using the guided notes that I've shared with you on the classroom. Use those to answer the questions and fill them in as you watch the PowerPoint, okay? And we'll go over these later. All right, we'll go over these in class tomorrow. Don't forget, I'm proud of you all. You're fantastic human beings, and I am lucky to have you as my students. All right, I will see you tomorrow, guys.